today.
Okay. Thanks. And I'm very glad to see several new faces, and most welcome. And hope these meetings will be productive. So I'm Olga. I am working at the National Genome Corps in Sweden. And what I'm going to share with you right now is the paper that we've done in collaboration with the Vertebrate Genome Project. So the background here is that in Vertebrate Genome Project, it's a very large project that has been going on since 2018, I would like to think. So they were one of the first projects under the EBP umbrella. And one might argue that's the easiest taxonomic group to extract DNA from, but at the same time, there were lots of questions. What's the best way to sample? What's the best way to preserve the tissue for the DNA extraction? And what extraction methods would result in the best quality of DNA? Taking into account different variables that one can encounter during the collection and in different taxonomic groups. So I came in this project quite late already. They were a year in, so the sample selection and the experimental matrix was done before my involvement. So this is a job that was run mainly by Hollis and Jackie and Jennifer. So these are the guys who were BGP partners, Hollis from Canada, Jackie and Jennifer, and also Giulio and other people. They are from the Rockefeller Institute. So they were acquiring the samples and doing extractions, and then sequencing was done at Sanger, also at Rockefeller, and in Dresden with Silke Winkler. So that's a little bit of the background on the paper. I came in the DNA quality assessment part because of the paper that I published on that. So this is a little bit of the overview of how the problem was tackled. There were 5 groups of taxonomic groups within the vertebrates. I'm not sure exactly what was the reason to include 2 different types of turtles, but I think taxonomically they were very different. But anyway, so what we were doing, we wanted to find out first if the flash frozen preservation is always the best, or can we find a second best, which is good enough, another way of preserving samples during the collection. So here you see a little bit of the matrix. So for the majority of the samples, we were using liquid nitrogen, fresh frozen, and then we were also looking for samples that were preserved without any buffer in any of these different conditions. So the samples were stored 4 hours at 4 degrees, and then samples were stored one week at 4 degrees. So it's not liquid nitrogen directly, but refrigerated. And then we also stored samples for 1 week at room temperature. And then we had the samples without any buffer, just as they are. We had ethanol, which is usually considered as second best to flash freezing. And then there was also DMSO, which is also a well-known preservative. And then we also used DNA guard. We also used RNA later for one of the samples, and also we did not run a massive parallel comparison, but we also used O-Protect for a couple of samples. So although the matrix was established, it was still very difficult to have exactly the same standard conditions for every taxonomical groups. So you'll see, for instance, that for birds, we did not have DMSO at that particular time, also because the tissue that was chosen was blood, and DMSO for blood, perhaps not the best way to preserve it. So that is a little bit of the matrix there, and you can see crosses where it has failed and did not result in any DNA. So from the tissues, we had blood. It also goes a little bit to the status of conservation of the species, where you cannot euthanize them, so you have a blood extraction on site. either from the captive individuals or from individuals in the wild. But where we could get some sort of the dissection, then, for instance, in mouse and in fish, then we also tested that on different types of muscles, like spleen here for the mouse, and also many different tissues for the fish. For the samples where we could do it, we could also work with muscle, but again, it was dependent on the tissue availability during the collection. But for these endangered species, we have collected only the nucleated blood, red blood cells. So here is a little bit perhaps difficult to see, but if you move closer to the screen, this is the result when we tried different temperatures and different treatments. So on the scales, you see that towards the right side of the spectrum, that's the proportion of the DNA that was not degraded. It was high molecular weight DNA. And there you can see that up, there is a fraction of 145 kbs plus. So something here we would consider as a degraded DNA. So what we have discovered at this stage, that the sole factor that was extremely important, that was the temperature. So if the samples were refrigerated at 4 degrees, after the collection, that was still quite fine. So you can see here, for instance, 4 hours refrigeration and then 1 week refrigeration and so on, 6 weeks of, yeah. But when the samples were stored at room temperature, everybody could see very strong skew towards the shorter fragments. And so that sample was more difficult to work with. But also we have found that in birds, the bird nucleated blood was extremely resilient. So in some of the samples, the bird DNA was okay, even 1 week at room temperature. But the best results still were achieved if the samples were pre-chilled before storing them at room temperature. For the bird samples, it was a heparin. So the blood was directly collected into the heparin tubes. So that is something that we learned. And we also learned that fishes were notoriously difficult to handle DNA. And also we have found out that muscle tissue is much more difficult to work with as compared to blood, spleen, and other organs. So we would not recommend muscle, especially if the cold chain cannot be maintained. So when we were testing different parameters, what is the most important of preserving this ultra-high molecular weight DNA. And when we say ultra-high, we decided to put a cutoff at 145. And the only reason for why we put this cutoff is because that's a detection marker in the pulsed field. So again, if we look here at the temperatures, when we have the flash frozen control, you can see that the room temperature at 37 degrees, where we wanted to mimic collection at the hotter climate, you can see that, of course, DNA was most degraded here. And then if it was in 5 weeks RT and so on, still you could recover something, and then 1 week at 4 degrees and so on. So it was quite a good recovery of the fragments, but still the flash frozen was absolutely the best. Again, when we were looking at the yield, how much DNA are we getting in the high molecular weight fraction, there was much more discrepancies. So that was most likely also sample specific. So we could not draw very sort of precise conclusions based on that. When we were looking at which fraction was over 145, of course, flash frozen or liquid nitrogen storing was the best. Also, we did not see any differences if you add any preservative or you just flash freeze without any buffer. So flash freezing without any buffer was a bit better, but not statistically significant. When we were looking at the mass of the DNA that we got all over, again, without any preservative, it was pretty consistent. But if we would get that in the preservative, so that would be the all over the chart. So again, it was pretty sample specific. And if we look at different tissues and look at the high molecular weight DNA fraction, again, there was no statistic significance. So it doesn't matter which body part you are using to extract your high molecular weight DNA. In terms of high molecular weight DNA, most likely you will get it. But again, the least spread was from the spleen and from the body. And then finally, corrected yield per mass. So from which tissues are we getting absolutely the best results? The most DNA you are getting from blood and from spleen and also from nucleated blood. And this is something that we knew from before. Perhaps in this particular group, it doesn't matter because not many of you, because I think only Jen is working on the vertebrates. But what we have discovered is samples with least connective tissue is usually the best, and spleen is a splendid candidate. If blood is not available by some reasons for euthanized animals. Another thing that we have discovered that very often after the quick euthanasia, blood drawn from the heart is a very good alternative. What was very worrying, because we didn't just check DNA extraction methods and also sequencing on the instruments, we also checked if different preservatives have any effect on the results of the high C, which is detection of the chromatin interactions. So for instance, here, that is how your high C distribution should look like. You should capture around 60% of the cis interactions, and you also are supposed to capture around 25% of trans interactions. So this is something that goes to the theory of high C. I don't know how many of you are familiar with that, so I apologize if I repeat some stuff. So high C tells you about the organization of the chromatin. You could use the high C information to scaffold that on your draft assembly and then define by proximity in chromosomes which contigs are sitting together. So this is the high C of a healthy individual. Ethanol did not vary too much from the high C. From the no preservative. So we conclude that ethanol is the second best option. And you can see that it's pretty much disregarding the majority of the samples. So blood stored as fresh frozen at 4 degrees, 1 week RT and so on. But when we were using DNA guard, and in a couple of cases also RNA later, we minimize the number of cis interactions and they're dropping dramatically. That means that the hi-fi data produced from the samples which were stored in DNA guard, and in some cases also in RNA later, will produce false results, so false positives, or will skew their results dramatically. In terms of muscle, we had quite a spread. So again, in flash frozen, it looks quite normal, so no preservative and ethanol, and RNA DNA guard still we have a little bit of the drop in the cis. But 1 week at room temperature, you could see what happened. So there you cannot utilize this data at all. And even if you refrigerate that at 4 degrees for 6 hours prior to freezing and shipment, still there was a little bit of, there were statistically insignificant drop in consistency in the production of the high C data. So finally, based on that, we have created a flowchart of which parameters one should consider when collecting vertebrate samples, where we postulate that the tissue type could play an important role, like for instance, spleen and blood versus muscle or ovaries that we also tested in a couple of individuals. Also the preservative solution, where we postulate that ethanol is the second best as compared to other known preservatives. Also, if we look at the short-term preservation, the most important is the conditions at which samples were stored when they were collected. So the higher the temperature, ambient temperature, the more degraded DNA was. And also, if the samples were refrigerated, even at +4 degrees, the results still were much better if they were stored at ambient. So even if people cannot have liquid nitrogen or dry shippers, having normal portable chillers already increase the chance of getting better quality of the DNA. And then also time in field storage. So where we collected this time series when there were 1 hour chilled or 6 hours chilled. So there still was a little bit of the correlation between the time when they were at room temperature and then how long they were refrigerated until they would come to a liquid nitrogen storage. Things like extractions and storage and transport, they play, of course, a very important role, but we did not have enough data to draw any conclusions about the shipment conditions. We ran a very simple shipment experiment when we sent samples on ice pack at room temperature and on dry ice, but we could do it only for one taxon. And yeah, the results were pretty much predictable, but we didn't publish that in this paper. So of course, DNA was much more stable when it was shipped in dry ice. Also here, perhaps I need to tell that we have compared different types of extraction methods, but because these samples at that time were also processed for bio-nano, when you need to really have ultra-long DNA fragments, perhaps in the megabase scale, the extractions that Jennifer and Jackie did was following the nuclei extraction, or casting the cells in the agarose plugs and extracting them in situ. So that would yield absolutely the highest quality of the DNA. And here I'm coming back to Jennifer's question. Do you really need to have ultra super high quality DNA? Answer is, what are you going to use it for? If you want to produce hi-fi data, or if you need to produce a data for Oxford Nanopore when you're sequencing bulk, which is fragments around 20 kb or up to 75 kb, then you do not really have to worry about your molecules that are over 1 megabase, because we will need to share the DNA anyway. And also, usually the normal degradation at this chilled DNA and so on, still we could recover quite a good peak of some of fragments around 145 kb, which is the fraction that we would like to work with when we're starting with PacBio sequencing. If, however, you want to go for the ultra-long nanopore reads, and those you will need only if you want to do some additional scaffolding, or if you are looking at the complex rearrangements on the supergenes and things like that, highly repetitive regions, then yes, you need to think about that. The samples must be preserved according to the best practices. But for the hi-fi sequencing, the main problem is actually purity of the DNA and not the structural integrity. If the majority of the fragments are above 60 kb and up to 145, you are fine. Because the shorter fragments can be eliminated. But again, the most important is that the fraction of the longer fragments is larger than the portion of the shorter fragments. So you should, what I'm trying to say, you should still aim for the high molecular weight DNA, but you don't need to worry about the ultra high molecular weight DNA. Hope it's clear. If not, ask me after. So, and then finally, these are the current VGP standards. Again, this is for the easiest taxonomic groups, for the vertebrates. So, and the tissue collection, we said that these is what we can work with, but again, we encourage to submit multiple tissue types, because we see quite a lot of variation between different taxa and between different samples. For amphibians, yes, also for fish, I can say that heart blood resulted in absolutely the best results, and one should try to avoid gills as much as possible, because the DNA from gills looks absolutely gorgeous, but up to 90% of reads can come from the contaminants. So amphibians, birds, non-avian mammals, non-avian reptiles, blood is absolutely the best way. Muscle is the second best because we saw higher levels of degradation in the muscle tissue. And in mammals, blood is always the king, but also spleen and muscle, if possible, should be used. But again, muscle performed a bit worse as compared to the spleen or to the DNA. And also we try to put different gradation on the preservation method, which is ideal, good, and acceptable. So, and that is flash freezing and short-term refrigeration before deep freeze. That is absolutely the ideal. So the shorter time it spends in a chiller before you flash freeze, the better for everybody. Good term or good one, it's ethanol. For the vertebrate samples, we did not have any negative impact on the high-C results, but I've heard from some plant people that ethanol does not work good in some of the high-C species. But again, it's a little bit anecdotal evidences, and we do not have a large data bank of sort of consequent parameters that we are checking. So it's very difficult to draw a global conclusion. And then acceptable blood and ethanol at room temperature was surprisingly good, especially for the birds. And that, I think, is everything that I wanted to share. So perhaps, Christian, if you would be so kind and put a link to the paper. I sent it to you earlier in the minutes, but I can also send a link to the paper here in chat. So if you want...
It's already in the minutes.
It's already in the minutes. Oh, cool.
Yeah.
Thank you so much. Thank you so much. So if you want to know more about the experimental design and how we assess different parameters, just go there. And we also have links to different papers that were looking at similar things.